And me and Raymond Roberts put together this symposium as a wanted to get just bioinformatics people, together to talk about Python and get a couple talks to inspire us and show some interesting things. Trying to do some academic and industry things. I think over the last couple days we've seen a lot of really neat applications of Python and scientific computing, which is of course one of my favorite reasons to come to this conference, is to see all the different things we're doing. And I know every year there's a different level of people doing bioinformatics here. Some years it's really big, some years it's really small. And we've kind of seen over the last few years really is there was how Python is becoming more important. So I wanted to kind of take a look back and talk a little bit about Python in bioinformatics and where it's been and where it's going, and kind of just have some discussion on that. So start off. SciPy, August 2006 in Pasadena. Was anyone there? This was basically in a cramped windowless room, a BOF convened to discuss the state of bioinformatics in Python. Was anyone at this other than me? You were there, so you can correct me if my history is wrong. But I remember kind of a bleak meeting. It just wasn't a very pretty meeting. It was a lot of discussions about, yes. Yes. Yeah, it was at Caltech, and Titus was driving a lot of the discussion. I was driving a lot of the discussion from a different perspective. And really, it was kind of a frustrating one because really BioPerl dominated the informatics landscape at this point. And we were really, it was kind of more of a sob session where we lamented this fact a lot over and over again. Bioconductor and R were starting to emerge as the Swiss Army knife of analysis tools. So we knew there was some competition in the space, and people weren't really turning to Python when they got bored with Perl or frustrated with it. And believe it or not, related at the same SciPy, there was a big discussion about web development in Python and what was actually important. And does Python have any tools that can really compete with Rails, which was really starting to become the dominant web platform at the time? And you'll probably remember at that time Zope was kind of our answer to all of that. I hope there are no Zope developers in here, but Zope was cool, but it was really hard to use, and it did frustrate a lot of people. So we kind of saw Python losing a lot of traction, not just in bioinformatics, but in the web space. And if you were doing bioinformatics in Python, and if you've been around Titus, you understand this, that bioinformatics projects in Python existed either by sheer force of will of their creators. They just were hell-bent on making sure that this was done in Python and that it was done Pythonic. Or there were also some knockoffs of BioPerl. I won't say much more about what libraries did that, but there were a lot of libraries that said we have to look like Perl to get the Perl programmers over here. So they threw away the Pythonic things and made their libraries look like Perl. So it was basically dark days for Python and bioinformatics. It was it was a fun meeting, but it was depressing, and it gave me a great slide to kind of show where we were at one point. So I want to kind of look at how that happened. Just go through kind of a history of sequencing informatics, and look at how different methods evolved. And this is really I did this on my computer. Now I'm showing it on Dan, so hopefully most of the graphics line up. But I want to look at kind of sequencing informatics, especially from the perspective of different tool sets that people have used, different ways that people have approached things, and it's really kind of meant to be more of a tongue-in-cheek thing. But I want to kind of look at and understand what's really changed and what's given Python some neat opportunities. So if we kind of look at bioinformatics, we kind of start off at the epic right around 1970. Believe it or not, 1970 was when Needles and Wunsch published their paper on global alignment. So that is one of the foundational bioinformatics algorithms that everyone I think still has to implement. If you take a bioinformatics 101 class, you either implement that or Smith-Waterman. These are basically sequence alignment algorithms. So if you have two sequences and you want to find out how similar they are, you can use these to align them against each other. And this really was the dawn of bioinformatics. Sanger sequencing evolved in the same time period. It was a nice, simple time. There were some basic first principles everyone was working with. The measurement tools were pretty basic. If you wanted to actually do sequencing, you were doing it in a lab and you were writing down the A's, G's, C's, and T's, pretty much by hand by just looking at gels and doing sequencing in a very simple way. So it was kind of a nice time. Our foundational data format, the FASTA format, came out then. Anyone who is in bioinformatics knows that we've been stuck with that ever since. And it kind of goes to say that sequence data is simple to represent, and we haven't really come up with anything that much better. Dot plots, were a popular way of looking at visualizing sequence alignments back in the day when you would only sequence 1,000 nucleotides. You could actually visually look at relationships between them, just as a simple thing. You're seeing the kind of homology here between the sequence on the x and y axes. So you could actually do a lot of analysis by hand. In fact, one of the early dot plot papers I tried to dig up for this, but I had a copy somewhere. They actually did it on a typewriter, and they used x's and dots, and it was actually pretty cool to see that someone published that back in the 70s. But that's kind of the antiquity time of sequence. If you were programming, it was using the tools of the day. C, Fortran were common. Couldn't find any punch card examples, but I'm sure there was sequence analysis tools written on punch cards at the time. There's kind of an example of sequencing more by hand there. So kind of after that, the Middle Ages of sequencing really started, and this was really driven by one major project. And we can argue whether or not it should be really called the Middle Ages, but we're going with Western civilization analogy here. Human Genome Project really dominated genomics, starting in 1984 when it was first proposed, going through the first 6 or 7 or 8 years of debating, should we sequence a human at all? What are the ethics behind this? Are we opening a big can of worms by doing it? And then really leading into the 90s where we did it. And went through and built some very large databases. It was really dominated by large sequencing centers. I would say the great databases, the NCBI databases, the protein databases, which we just saw being used in the last talk, and really what's the foundation of a lot of what anyone is doing sequencing now. These were all built. So the NCBI and European version and the other various national versions of these great genomic databases, where basically our knowledge of genomics is captured. And around the same time, in 1996, was kind of a pivotal event for Python or any programming language in sequencing informatics. And this is where Lincoln Stein gave a talk. I've got his picture right there giving the talk, about how Perl saved the Human Genome Project. And his main thesis was that you had a couple different sequencing centers all doing basically the same thing, and having to share data that was in slightly different formats. And it was really by injecting Perl into there and making it very easy to convert data back and forth between different formats and kind of standardize some of that interchange that the Human Genome Project was actually possible. So he basically pushed that thesis, and it was actually true. I mean, there were a lot of data interchange problems, and it was kind of a mess at the time. And going to scripting languages and doing what we kind of have all done, doing scientific software development, we're all very familiar with this, where you have to do a lot of conversions and a lot of pipeline development. Perl was the language of choice, and if you were around sysadmins kind of in the mid-90s too, kind of how that happened is that's what everyone was using for text transformations and parsing log files and data conversions. So it was kind of a natural choice. It did have the unfortunate effect of everyone read that as, if you want to do bioinformatics, use Perl. So we have what I'll call the Perl monoculture that started at the time and really lived for a long time. And this is probably the single biggest event in preventing Python from growing, or any other scripting language for that matter, from gaining a strong foothold. At the same time, we did see some amazing science and computer science and bioinformatics being done. I think BLAST actually, I think, is one of the coolest research projects ever, because it's really a project that was taken on as the intersection of statistics, biology, and computer science. So you had strong components in every one of those. You had to develop the statistical models to understand the differences between the different genomes and come up with a way to basically be able to search. So BLAST, if you're not familiar, is the tool you use to search it was the original tool that you used to search for sequence similarity across databases. So if you had some gene that you just sequenced, you would blast that against the human genome to see where it matched. But it required a lot of statistics work, a lot of working with the biologist to understand how sequences can evolve over time. So the way BLAST actually works is there's a lot of it uses a statistical model that allows for substitutions inside the nucleotides that you see, or the peptides if you're dealing with proteins. And a lot of there was a lot of domain knowledge put into there, but then also a lot of methods for building up those statistical models when you don't actually have all the domain knowledge or all the evolutionary knowledge. So a lot of methods that just how do you take and make a great computer program that uses statistics, domain knowledge, and also has to actually work at scale and build that. So that's kind of why I always think BLAST is one of those great computer science or great interdisciplinary projects. And there were some other programs. Hidden Markov models were really popular at the time, especially for predicting protein structure and domains. There was a lot of work there. And multiple sequence alignment is one of the foundational methods for doing comparative genomics, where you have really large genomes that you try to align against each other to see how similar is the human to the rat genome or other ones, one use case there. So you had some great tools that came out. A lot of them were written in C, and then a lot of the glue code was, of course, now starting to be really written in Perl. So moving on, we kind of hit a weird period in bioinformatics and genomics in general, the dark ages. And really, the highlight was the human genome was published. This was actually an amazing thing. The draft was published in 2001. The final references were published around 2003. But at the same time, there was this huge realization that if we want to do genomic medicine and all of the promises, all of the things that we were promised with the human genome, then we had it sequenced. We couldn't just sequence one person. We would have to sequence everyone. So there was kind of this big collective sigh and a great sucking sound as all the investment put into genomics was sucked out of it very fast. A lot of my friends worked at a genomic informatics company at the time, and the investors, within a period of 6 weeks, took all their money back and shut the company down. And that happened across the board with a lot of biotechs and a lot of companies that had basically bet their future on the human genome being this great tool for genomic medicine. And it was really just that realization that we've got to do this for everyone, and we don't have the technology to do it. The human genome, the one genome we got was a $3 billion project to sequence essentially one person. And some people might be worth $3 billion, but not everyone. So it's just not going to happen. So we entered kind of a weird period. For people who were still doing research on this, BioPerl really evolved then. BioPerl provided access to all the databases. So a lot of people were just writing their tools in BioPerl. And that was kind of the end of bioinformatics right there. A lot of bioinformaticians were even looking for other jobs at the time. Java was starting to gain some foothold in the language space too. Again, if you were in academics at that time too, you started to see everyone trying to rewrite everything in Java because that was what industry had just done. So it had trickled over to academia. Not the greatest time. There wasn't a lot of positive things in there. But through all that work, there were some rays of light coming through. And we did see certain things. So in sequencing the human genome and developing all those methods, a lot of other things were actually sequenced at the same time. So comparative genomics, and these are all fields that this field had been building up throughout the time. But this is when really you started seeing a lot of comparative genomics papers coming out, some really neat applications of it. This picture right here I have is from the VISTA project at Berkeley Labs, where they basically, it's one of the neater projects that they did. It was just kind of a parameter sweep on the embryos here, and they basically just went and tweaked a bunch of cells systematically. So they basically had people going and growing these chicken embryos and taking pictures of them at certain times and watching what changed based on gene expressions, based on what they had changed. They didn't go in there with a hypothesis. They just went in there and did the parameter sweep. So they had 40,000 embryos that they had these pictures of going afterwards, saying they're going to go through and mine that data and try to understand how the gene pathways actually work. So these are the types of ideas that, when the Human Genome Project fell apart, people started to say, well, what else can we do with all of this technology that we've all just bought? We have the supercomputers we were using. And there was a lot of neat stuff going on in this space, trying to figure out how do you actually do these bigger genomics. Sequencing RNA, which Dan talked a little bit about. ESTs was kind of an older way of doing that, of sequencing the express sequence tags. So taking the RNA, turning it into DNA, and then kind of seeing what it is, which is still kind of how we do RNA sequencing today. Microarrays became really popular. So there was stuff going on, and this was really starting to show in bioinformatics that there were a lot of applications. And more importantly, a lot of these applications were starting to become more and more computationally intensive than what people had done before with, the human genome was computationally intensive, but afterwards just doing simple BLAST queries was basically a solved problem. So there wasn't a lot of interesting computational stuff to be doing at the time until you start doing the analysis and doing some of these bigger experiments. Along the same time, a lot of the annotation databases, people continued to contribute to them. So if you look, the ones that Adam mentioned are some good annotation databases. Another one is the SNP database that shows where all the variations in the human genome have been discovered. Really, the end game for that database is every position in the genome. But over time, we've seen that database evolve, and it really kind of started to fill up at this point, especially when some of the big sequencing companies went out of business and just dumped all of everything that they had learned into that database. So Celera, for instance, all of the SNPs that they had discovered and that they had intended on keeping private and making money off of, when they decided they couldn't, they just contributed them all back. So we got some really large databases. So we started to see these really large data collections build up, and a lot of people finding more things that you could do with genomics. BioPerl, still around, not going anywhere. And R and Bioconductor. So Bioconductor, this is, really I think Bioconductor was 2002 or 2003. Sometime in that time frame is really when it came out and started to get hold. And it's an R package for doing a lot of types of analysis. this would have been a great chance for Python to jump in, but Bioconductor, they chose to use R instead. So, and that's really right there is kind of the root of R in bioinformatics. Is Bioconductor is kind of a catch-all for all the analysis that you would like to do in bioinformatics. So there's a lot you can do by just firing up R, loading Bioconductor, and pulling in your data. So you still see a lot of Perl, Java, and R in the space. So this catches us up kind of to where we were in 2006 with Python, trying to figure out, all right, we've just watched the whole field of bioinformatics develop. We've seen one cycle of crashing. We're starting to see more cool applications, but no one's using Python outside of these few niche areas. Is there anything we can do here? It was bleak, but as this picture shows, there was a little ray of light in the middle, and kind of not even just metaphorically, a lot of this ray of light came from this new type of sequencing that was being introduced. So if you remember with the Human Genome Project, it crashed. That whole field crashed basically because we couldn't do this for everyone, which is what they really wanted to do. So we had to find a way to take that $3 billion and get it down to the $1,000 genome that we've all heard about on the news for probably the last 10 years. That's been mentioned a lot. And what happened in 2006 is a company called 454 released their sequencing platform that started to enable this. So it wasn't a $1,000 genome at that time. It was more of a $100,000 genome. But what changed is that what took billions of dollars and a decade to do, you could now do in about 2 weeks of time for about $100,000 on an instrument that cost $500,000. So a well-heeled lab could now suddenly start doing real sequencing. The catch there, of course, was that sequencing 3 billion base pairs for an individual person, you have to actually sample fairly deep to do this just because of errors in the instrument and errors in biology. So it generates a lot of data. So now we're not just looking at 3 billion base pairs in the human genome. You're looking at 3 billion times 15 or 30x or 100x, depending on the experiment you're doing. So suddenly you're collecting tens to hundreds of gigabytes of data just to ask a single question. So of course, the first thing everyone realized is all of these tools that we've developed don't scale to a billion reads. Reads are the things that come off the instruments. And there was suddenly a chance basically to turn the crank on bioinformatics and redo all the tools to focus, to be able to work at scale. So at the scale that we're doing, to do actual human genome sequencing kind of on a 2-week time scale or 2-month time scale as it was at the time. Kind of shooting for getting that cost down from $100,000 to $10,000 to $1,000 to where it's going to be. And as the green coloring here is actually where Python starts to show up in a fairly major application. So 2 of the other platforms, so there were 3 platforms that kind of all came out around the same time. There was the 454 system, the solid system, and then the Solexa system. And the solid system came with this whole pipeline building kit, and that actually was mostly Python. So it was called Corona. And did anyone here use Corona when it was? Or use Solid? Did anyone use Solid? But okay, but so a lot of their bioinformatics tools were actually Python, and a lot of their pipeline management was Python. So that was kind of hidden behind the scenes. Not a lot of people knew it, but there was a huge code base that was actually developed in Python specifically for sequencing, and kind of seeded the idea, at least inside Applied Biosystems at the time, and LifeTech, where Dan is now. So Python actually developed there was a decent bioinformatics culture developing internally in there, so you started seeing more people using Python in this space. What was kind of exciting about it too is
open source, or was it proprietary?
it was pseudo open source.
So it was like available.
If you bought the instrument, you got it, and then if you actually looked at the license, someone inside had stuck the GPL on top of everything. So it technically was open source. I won't go into many more what I know about that, but yes, it was. And no, none of the management knew it was, so. Or appreciated what that actually meant when they dropped that license on it, so. But yeah, it was kind of open source. But kind of like BLAST being this great chance to do a nice interdisciplinary project and bring together computer scientists and bioinformaticians and statisticians and biologists. We had this opportunity again, kind of came about when next generation sequencing came along. So the first thing that people really tackled was the aligners. So BLAST is a great tool, but BLAST just can't map a billion reads against a reference in any reasonable amount of time unless you have a blue gene or something like that available to you. And people have done that. So, but not everyone can buy one of those and maintain it and write code for it. So the whole the whole set of alignment tools had to be rewritten from the ground up to start to work with the data as we were seeing it. So what kind of changed here is the data is shorter, so we're mapping short reads now, so things that are kind of under 500 base pairs to things that are 3 billion base pairs long. So there are some optimizations that you can do when you're doing that. They also, for the early ones, actually for most of them, just to get performance up, you also threw out some of the things that biologists like to see. So for instance, when you're aligning sequences, you're always going to have mismatches between your reference and what you're sequencing. There is variation in nature, and there's error in the instrument. So an aligner has to be able to do that. But one of the variations that happens is you'll have insertions and deletions. So your reference might have 4 bases missing in there. So all of the short read aligners decided to ignore that because it made them work fast. And you could say computer scientists did a great thing. But really what they did is they just kind of ignored a huge source of variation. And so some of the newer tools now are starting to bring that back in. But biologists,
Short read won't allow you to do that.
yeah, they will.
Not adequately for you cannot because there are insertions.
Yeah, you can't see them all, but you can see a 1 or 2 base insertion or deletion. So small insertions, so you can't do large indels, but you can do small indels. And then if you're dealing with a platform like Ion Torrent, they're close to 400 base pair reads now. So you can do 10 or 20 base indels just fine on there. So that was all, and that was one of the things is that for that reason too, and for a lot of reasons people didn't think it mattered, but it did. And so what's kind of exciting there is it's a chance to rewrite the software stack from the ground up. And again, a lot of this was done in C. There were some new formats. FastQ is kind of replacing FASTA now. It adds one more line, one more bit of data in there, plus a line that's meaningless. So it's kind of one of those weird formats. And then some other new formats that came along. But you started to see now more tools being used. And a lot of people coming from other scientific disciplines coming into bioinformatics now. So people like me coming from doing other types of scientific computing and saying, look, Python's is a great language for tying a lot of this together. And what we started to see with the tools coming out are a lot of the libraries are C libraries now, just because of the scale of data we're working with. A lot of the stuff is written in C under the hood, and people are putting language bindings for all the major languages now. So we're starting to see a lot of Python bindings, and those, even more to the point, we're starting to see a lot of people choose those when they're writing their pipeline. So they're actually doing their pipeline development now in Python and just using these libraries to actually do the data processing. People still use R for a lot too. So R is actually bioconductor is probably the dominant player here, but we are seeing a lot more with Python. So in the last couple of years, we've really seen now what's the equivalent of the industrial revolution in sequencing, to where we're seeing true industrial scale genomics. There's a company, Complete Genomics, that is basically their business model is how many human samples can we push through in a month to do cancer sequencing applications. The Broad out on the East Coast has a huge sequencing center set up, and they are just pumping out sequence after sequence after sequence for people. You submit your samples to them, and you'll get a lot of neat genomics done on it. The real big one, of course, is BGI, which is the Beijing Genomics Institute out in China. This is kind of the socialized capitalism model of if you pour $3 billion into a company and hire an army of people. Apparently, they have 2,500 bioinformaticians just working at the BGI. And the person who told me that was the person who was managing them all, who was 26 years old. So it was very different than the way we would do things here, but there you have it. So very, very much industrial scale sequencing, and they are just cranking out a lot. It's most of the publications you see now of the new genomes being sequenced are genomes that were sequenced at BGI, and this has really been enabled by all of the low-cost sequencing. Another one that I guess got lost on my slide is, oh wait, that's the next slide. Never mind, sorry. So instruments are also commoditized. So you can buy your sequencing instrument from anyone, and you get the same basic type of data out of it. And now we're seeing a lot more libraries and toolkits. Just as any field matures, we start to see a lot of the abstraction layers being lifted up higher and higher. So a lot of these are coming out. HT-seq is a popular one right now, and that actually is a pure Python sequence analysis package that a lot of people use. And another really interesting one that there was a talk here either last year or the before is Galaxy, which is kind of a whole pipelining engine and application layer. There's a little screenshot down there. But it's a web application that you can use to do a lot of your sequence analysis with. And that's actually really taken off in the biology community. Biologists like it a lot because they don't have to use a command line to do their analysis. And it is right now the best kind of closest thing to a GUI for sequencing out there. Still not perfect, but it's definitely a step in the right direction. It's a Django app, so it's pure Python. It's actually just an egg, so you can drop it on your workstation and run it if you want to run your own local copy, or you can install it on your clusters. They've done a really good job of making it very easy to install, and I think as much as we hear people complaining about Python installers, they've actually done a good job of making it easy to install stuff for kind of a non-Python user to just drop it in and do it. So that's kind of an exciting thing for Python in this space. So now we're getting into what I'll just call, because we're running out of Western civilization analogies here, just the genomic age here. And the cool thing that's really happened in the last year is the introduction of benchtop instruments. These are instruments that don't cost $500,000. They cost around $50,000 to $100,000. They take about 2 hours to run, and the newer ones that will be kind of coming out this summer can do an entire human genome in about 2 hours for about $500. So, we're kind of going past the $1,000 genome into the $500 genome, ignoring the $20,000 of bioinformatics you have to do to get any answers. But as far as reagents, and basic sequencing cost, we're down. We've passed that point to where we can actually sequence people fairly quickly now, and that's just totally democratizing sequencing, especially large scale, like human scale sequencing. So not just looking at small species that were fairly easy to sequence, but now we can do a lot of mammalian sequencing, or just go sequence everyone in this room and find out what's different between us. Which of course leads to where a lot of people are wanted this to go with the human genome projects is into the clinic, so clinical genomics, and you kind of have a picture here of showing what clinical genomics might look like, and you're going to have a very confused scientist or doctor trying to make sense out of a lot of this genomic data. So there's a lot of work to be done in that space to figure out how do we actually interpret this and how do we put the tools in place. But there's also a lot of opportunity here now for people to develop new tools in new languages, or more importantly, new tools in Python. So there's a lot of opportunities here. We're seeing lots of libraries and applications coming out, people deploying these on public and private clouds. As you can imagine, this is a real big data problem. And that's not being that's not hyperbole. So if the new ion torrent instrument that has kind of been announced will be generate that can do a human in 2 hours. That's about 500 gigabytes of data that you get dumped on you every 2 hours that you would have to process if you're doing this. So if you're running a few of those, you can easily fill up a fairly large storage system, as I know that they have inside LifeTech. So they're past the petascale of filling that up fairly quickly. So it's real big data, a lot of processing you have to work with. So there's a lot of managing that data and everything else that goes along. So if you've been watching the SciPy talks for the last 10 years, you're starting to see, with sequencing, what I see is the parallels that astronomy and high energy physics and a lot of the other people have had. It's the same problems are starting to come into bioinformatics now, and we just need to basically take the best ideas from there and very quickly get them out to bioinformaticians. And I think there's a lot that's been done already in Python that we can leverage. And of course, Python's here. I have Ion Torrent highlighted there too, because their entire instrument control software and their informatics pipelines are all done in Python too. It's another Django application with a lot going on. So it's coming along. So, and then in 2038, we all know what happens. Time ends. So it's not the Mayans in 2012. It's 2038. So really, Python, it was a late bloomer in sequencing informatics, but starting around 2006, when we had to rebuild the infrastructure from scratch, Python, finally gained a nice foothold. And I think a lot of what we've learned, doing other scientific apps is directly applicable here. So I think Python's got a great future, along with other languages in bioinformatics. But I think Python's going to be a big one. So if you're a scientist not working in bioinformatics and your bioinformatics friends, ask you what to do, definitely push them in this direction. I think there's a strong enough ecosystem now out there that people can do this. So, thanks.